In this lecture, we will discuss about the hybridoma technology for the monoclonal antibody production.
It is simply a method to produce monoclonal antibody specific to antigens of interest. And this technology is developed by Kohler and Milstein. They got Nobel Prize in 1984 in Physiology and Medicine for the discovery of this technology. Monoclonal antibodies are set of groups of identical antibodies. They are derived from a single clone of activated B cell. These antibodies can only recognize a single specific epitope on a target antigen. Here is the yellow colored monoclonal antibodies are recognized only this yellow colored epitope on the antigen. That is the basic idea. But in the case of these polyclonal antibodies, they contains a mixture of antibodies and they are derived from a multiple clone of B cells and recognize different epitope on the same antigen. That is the main difference between the polyclonal antibodies and monoclonal antibodies. Next, I will explain the major steps that involved in this hybridoma technology for the production of these monoclonal antibodies. First step in this hybridoma technology is the immunization of rat and extraction of B cells. First we require a rat and this rat is immunized with the antigen of interest. What happened here that these antigens are enters into the circulation of the rat. Then it directly enters into the secondary lymphoid organ in the rat which is called as the spleen. When these antigens are enters into the spleen and these antigens are recognized by the B cells in the spleens and get activated. That is the next step, the extraction of this B cell from the spleen. So we remove the spleens after 72 hours of immunizations and it is macerated into several fragments. From the fragments of tissues we can isolate the activated B cells or the antibody producing cells by using several kinds of enzymatic method. Second step is the isolation of the myeloma cells. Myeloma cells are the fast growing immortal cancer cells. After the extraction of this myeloma cell and B cells, next step is the fusion between these cells. That we require the B cell and here is the B cell. Also we require the myeloma cells. For the fusion we require a fusing agent. The fusing agent is done by this polyethylene glycol. Now what here done is that this B cell and myeloma cells are transferred into this medium that contains the polyethylene glycol. The polyethylene glycol induces the fusion between these cells. At the end we get 5 different kinds of fusion product. One we get fused B cells, also we get fused myeloma cells, also we get fused B cell and myeloma cell, that is called as the hybrid cell. In this case it is called as the hybridoma cells. Also we get unfused B cells and also we get unfused myeloma cells. Remember, B cells are the antibody producing cell, also the myeloma cells are immortal in nature. When they are fused together to form the hybridoma cell, that they get the both characteristic feature of this B cell and myeloma cell, that is hybridoma cells are antibody producing cell and also immortal in nature. Fourth step is the selection of hybridoma cells. Here is the mammalian cells. The mammalian cells can synthesize nucleotide by two pathways. One is the de novo synthesis, second one is the salvage pathway. Also, the selection is done by this HAT medium. HAT is for the hypoxanthine aminopterin thymidine medium. Aminopterin block the de novo synthesis, so mammalian cell cannot synthesize the nucleotide in the HAT medium. Only the salvage pathway is available for this mammalian cell to synthesize nucleotide in the HAT media. For the selection, the culture medium containing fused and unfused cells is must transfers to another medium, that is actually the HAT medium. The key idea behind this selection is that the B cells contain a gene which is called as the HPRT1 gene. They code for an enzyme which is called as the HGPRT for the hypoxanthine guanine phosphoribosyl transferase. It is the key enzymes that helps in the nucleotide synthesis by salvage pathway. We already learned that mammalian cells can only synthesize the nucleotide by salvage pathway in the HAT media. So the B cells can grow in the HAT media. But few days after they will disappear because the lifespan of the B cells are very short. So they will disappear. And second one is the myeloma cells. The myeloma cell do not contain this HPRT1 genes. So the HGPRT enzyme is don't work. So that they can't grow in the HAT media. The remaining is the hybridoma cells. They are immortal cells. Remember, these hybridoma cells are formed by this fusion of this B cell and myeloma cells. So they get the HPRT1 gene from the B cells. So they possess HGPRT enzymes, that is the hypoxanthine guanine phosphoribosyl transferase. So they can grow in the HAT medium. So because of this immortality in nature, they can grow in the HAT media for indefinite time. So after few days only the hybridoma cells are remain in the HAT media. In this manner we can select the hybridoma cell very easily.
The fifth step is the screening of hybridoma cells. Let's imagine this is the antigen that is introduced into the rat during the initial stage of immunization. This antigen possesses different kinds of epitopes. We can see that this yellow colored, blue colored and green colored epitopes. When these particular antigens are enters into the rat's body, it is directly enters into the spleen, that is the secondary lymphoid organs. When it is enters into the spleen, these antigens are recognized by the B cells, and the B cells get activated and produce antibody against these different kinds of epitopes on the antigens. That we get three kinds of antibody. The yellow antibody bind to this yellow epitopes, that green bind to this green epitope, and blue also bind to this corresponding epitope. That we get actually different clones of B cells that actually producing different kinds of antibodies. From this we only require one kind of antibody, that is the group of identical antibodies actually called as the monoclonal antibody. Let us imagine our antibody of interest is this yellow colored antibody, that we requires this yellow colored antibody producing B cells, that is actually called as the single clone B cells. For the selection of this single clone B cell from this mixture of these hybridoma cells, use a method which is called as the ELISA or RIA. By using this method, we can isolate that particular single clonal B cell separately. Next step is the culture of hybridoma cells. During the initial stage, we only get few number of hybridoma cells. For the production of large number of monoclonal antibodies, we require large number of hybridoma cells. It is actually the multiplication of the hybridoma cells, that it transfers to another medium that contains insulin, transferons, amines and other hormones. By the help of these supplements, these hybridoma cells multiplies and proliferate and increases their numbers. Some other commonly used culture media in this monoclonal antibody production is that HARMS F12 and RPMI 1614 medium DMEM. After the culturing of these hybridoma cells, the sixth step is the inoculation of hybridoma cells into the suitable host for the production of the antibody. Now what happening here that these hybridoma cells are injected to mouse. These hybridoma cells get multiply and produce a fluids in its abdomen of this rat, that is actually called as the ascitic fluid. This particular fluids contains a high concentration of that specific antibody. Here is our yellow colored antibodies are produced in the ascites fluid. Or these cultured hybridoma cells can be frozen in the liquid nitrogen for the future use. Last step is the extraction and purification of the monoclonal antibodies. Now our hybridoma cells are produced the monoclonal antibodies within the ascites fluids of the rat. Now we want to remove that ascites fluids and extract that particular monoclonal antibody. There are several methods are used for this purification of these antibodies are ion exchange chromatography, antigen affinity chromatography, immune precipitations are also used. After this precipitation and purification method, we get a pure clone of identical antibody that actually binds to a specific epitope, which is called as the monoclonal antibody. These are the steps that involved in this production of the monoclonal antibody. I hope you get an idea about this monoclonal antibody production by this hybridoma technology. If you like the video, make sure to subscribe my channel, hit the bell icon for the instant notification for the future update.